why don't we go ahead and get started? So we have sort of a two-part presentation. One is to sort of following, in both of these things obviously follow on the talk that I gave earlier today. But one is to talk a little bit about the considerations for the experimental design, and to describe how we got to where we did with the experimental data generation, and then to give a quick run through of where those data currently live. And with that, I will hand over first to Sean.
Perfect. Can you see my slides and hear me?
Yes.
Yep. Okay. So I'm going to talk about the MCF10A project, which is in kind of the decision points that went into the design. It's never straightforward after the data comes out, how we actually got to the point of testing what we did. So I kind of wanted to cover some of the details of that. So for people who are analyzing the data, you can kind of understand some of the decision points that were made. First of all, just a brief overview of the MCF10A project. So the general goal of this was to first try to identify some high impact ligands, so ligands that alter the phenotype of cells, begin to quantify the responses of those cells to the different high impact ligands, measure a number of different molecular phenotypes using links assays, bring all that data together to integrate it, and then begin to identify patterns within there and modules to begin to relate the different phenotypes to the molecular correlates. So as part of the project, we use 7 different conditions. So the ligands TGF-beta, BMP2, interferon gamma. We also use EGF, oncostan M, and HGF along with a PBS control. We collected assays at different time points. Not all assays were collected at these time points, but we collected data at 0, 1, 4, 8, 24, and 48 hours. And we collected a series of different assays, including RPPA to identify changes in protein content and phosphorylation, cyclic IF, which stands for immunofluorescence, so we can begin to track the behavior of the cells, RNA-seq, L1000, both transcriptome-based readouts, and GCP and ATAC-seq, which are both kind of epigenetic readouts. And the idea is we can begin to track these signals from these high-impact ligands through the different types of biological information, including changes in protein phosphorylation through signaling, changes in transcription, changes to the epigenetic landscape of cells, including changes in protein and behavior. This was a multi-center experiment. Much of the cell culture was occurred at OHSU, but we also had help from other institutions, including MDACC, who ran the RPPA samples, Clemson, and specifically Mark Birtwistle and Jamal, who helped with analysis of the data, Mount Sinai with data coordination and analysis, and then MIT, Broad, and Harvard specifically in helping with the cyclic IF data. So in constructing this experiment, there were a number of decision points that came up that we thought about in depth and tested quite a lot to try and make sure that the data was as robust as possible. And what I'm going to do in this presentation is begin to kind of work through each of these different questions here and how we were thinking about those questions and trying to design the experiment. So the first question is what cell line to test. Second question, what media to use, which turns out to be quite complicated, what ligands to test, what time points, and then how to split into different batches. The general take-home message I'm hoping is that the people who are watching this can get a rationale for the different methodological decisions that we made. In addition, that there are likely to be future experiments and likewise build-outs from this data set. And so hopefully this can be helpful for other people as they go through and try to design these sort of experiments. So the first question is what cell line to test? And there were a number of kind of decisions that went into this. First of all, we wanted it to be a relatively well-studied cell type, specifically so we could begin to relate to other data within the literature. In addition, we wanted a cell line that is relatively genetically normal, specifically diploid. And one of the important things, especially when we're trying to relate phenotype to molecular correlates, is that we wanted the cell line to be relatively dynamic. And so specifically a relatively short doubling time. So the the MCF10A cell lines have a doubling time of approximately 24 hours. In addition, they're quite dynamic in their movement. So I can show a movie here kind of showing the cells as they go about migrating across the plate. And you also see the frequent division of the cells. And so in trying to begin to understand what the molecular correlates of this behavior is, it is very helpful to have this sort of behavior for a cell line. One of the additional criteria, as shown in the movie, is these cells are largely growth factor dependent for their proliferation. So for example, on the right in the movie, we're showing the addition of EGF, which is driving the proliferation. As we show throughout this presentation, other ligands can potentially drive this proliferation, but it is an important factor for these cells. So they don't necessarily have an oncogenic mutation that is driving their proliferation independent of any sort of external stimuli. One of the potential trade-offs within any sort of cell line is the media. And this was one of the big trade-offs in deciding to use MCF10A as a cell line because they have a relatively complicated media. And so in doing that, the question is, how do we adjust the media to be able to identify the effects of different ligands? So media in general is designed to promote the proliferation of the cells. So their cells are really dividing relatively quickly in response to the media. If we're trying to look for ligands that induce proliferation, we need to try and kind of correct the media so that we can begin to identify those effects. So the MCF10A media itself is composed of a 50-50 mixture of DMIM-F12. In addition, it has a number of different additives. So it has pinstrep, hydrocortisone, cholera toxin, horse serum, EGF, and insulin within there. So our first approach to try and reduce the proliferation of the cells was to dilute the media. So begin to mix in DMIM-F12 to lower the percentage of all the other additives within here. It's a relatively straightforward approach. And we were able to do that. So we were able to do it, reduce kind of the media concentrations of all these, identified an IC50 concentration. But one of the things we found is that there was no EGF-induced proliferation under these conditions. So we still, the media was still covering up the ability of the cells to respond. And so then we engaged on the second approach, which was trying to do a little bit more of a targeted approach where we go about removing individual media components. We tended to kind of group these together in terms of the hydrocortisone and the cholera toxin. Horse serum stood on its own, and then also EGF and insulin together. And we went about doing this and removing each of these individual components. You saw very different effects on the proliferation. So in this conditions, which I'll show here, so if we keep the horse serum within, but we remove the growth factors, so we remove EGF and insulin, keeping the hydrocortisone, you see proliferation is significantly reduced. On the other hand, if we add back EGF, we see a robust proliferation close to what's the cells do under the full media conditions. Then when we went about removing other different components, we were never able to get the same sort of effect in terms of EGF-induced proliferation, which is a control. So if we removed the hydrocortisone and the cholera toxin, you do get some EGF proliferation, but the dynamic range of these is much reduced. Likewise, if we remove the horse serum, but keep in the hydrocortisone and cholera toxin, we get very little EGF-induced proliferation, as well as when we remove both the horse serum and the cholera toxin. So kind of we ended up with this final MCF10A experimental media where we went about removing EGF and insulin. And so with this, we should be able to identify then any effects from the ligands to be able to induce proliferation. Likewise, since we get close to near maximal proliferation just by adding EGF, we can identify ligands that reduce proliferation in the context of EGF-induced proliferation. So after deciding on the cell line to test MCF10A and modifying the media in order to see an induction in proliferation and also a reduction in the cell number, the next question is which ligands to test. And to do this, we tried to do a knowledge-based approach using receptor RNA-seq, which is both our lab has collected and then also many other labs have collected. And what we did is we developed this kind of idea of the receptome, which is based out of Kevin Jane's lab in a paper there from 2013, where they went about identifying different classes of receptors. So the cytokine receptors, TNF, TGF-beta, chemokine, this kind of other group, which includes Notch and Gli and Toll receptors, the receptor tyrosine kinases and nuclear kinases. And so we can tabulate these. And one of the key things about these different classes of receptors is there are canonical downstream signaling pathways that are activated by these. So for example, the cytokine receptors predominantly activate STATs. They're able to activate potentially other downstream signaling pathways, with the primary driver are STATs. Likewise, with the TNF receptors, NF-kappa B, the TGF-beta BMPs are largely driven by the SMADs. Chemokines are largely G protein based receptors, and then the receptor tyrosine kinases largely drive ERK and AKT activation. And so we could go about doing that with MCF10A cells. So using the RNA-seq, so we have expression across the x-axis, and we can then group these receptors into different classes and identify what are the highly expressed receptors and what are the lowly expressed receptors. So in order to try to identify ligands that have high impacts, the receptor needs to be expressed. And ideally, it's relatively highly expressed. So that's likely to see most of the cells express it rather than a fraction of cells. In addition, it allows you to identify less commonly studied ligands. So for example, if we look over in the cytokine receptors over here, we see that the oncostatin M receptor is one of the highest expressed receptors within there. Likewise is its co-receptor, the IL-6-ST. Very little is known about oncostatin M, but the receptor itself is highly expressed, suggesting there's likely some sort of phenotypic effect of it. Likewise, if we look at interferon gamma, the two co-receptors are two of the top expressed receptors. So in general, what we tried to do then is we looked at the receptor list, tried to pick receptors which have soluble ligands we can go about adding to the cells, and then we chose to pick two different ligands representing the cytokine, the tyrosine kinase, and the TGF-beta BMP2, with the idea that these are going to activate similar but slightly different downstream signaling pathways between the two paired ligands. So interferon gamma historically activates more STAT1, whereas oncostatin M is thought to activate more STAT3. EGF and HGF, so EGF tends to activate ERK a little bit more than AKT, whereas HGF tends to sway in the opposite direction. Likewise, with BMP2 and TGF-beta, BMP2 tends to activate SMADs 1, 5, and 8, whereas TGF-beta are SMADs 2 and 3. In addition, with trying to come up with these ligands, we did several validation studies and also tested other ligands to go about validating the MIMA results that we use as part of the information to decide on these ligands. So after identifying the ligands, doing some preliminary tests, the next thing we did was dose response of these. So one, it validates that these ligands are actually, modulating the phenotypes that we're looking for. So here what we have is we have ligand concentration across the x-axis, and then we have kind of a normalized cell count, normalized EGF across the y-axis. And you can see here that as we increase the dose of each of the three ligands, we see a clear dose response pattern within here. We chose to test for the MCF10A paper doses that were above the maximum value, with the idea that we're less likely to have variation if we tested doses at the IC50 dose. And then you're also less likely to have depletion in the media across time. So in tracking EGF at submaximal doses for 48 hours, we tended to see reduction in the effect as the time of the experiment extended. One of the things that we had to do is that we, in the three ligands, the reduced cell numbers, so the antiproliferative ligands, interferon gamma, TGF-beta, and BMP2, we were doing a time course experiment which required us to collect samples over 48 hours. And one of the things is that ideally we would have been able to test these on their own, but in order to collect all the necessary samples, we had to add EGF in order to get a proliferative signal so we have enough material at the end. So in experiments we've done where we're just adding these ligands on their own, we end up with almost no cells at 48 hours. So even in the absence of EGF, they're still reducing proliferation. But if we end up with no cells, it works for IF and in terms of quantifying the number of cells, but it doesn't provide any material for RNA-seq RPPA, GCP, and the other assays that we collected. In addition, besides the 6 ligands talked about previously, we also collected a PBS condition. And one of the things in analyzing the data is that it's helpful to have a variety of different controls. So it's important to have kind of time-based controls. It's also important to have a ligand-based control. So we had the PBS, phosphate buffered saline, as kind of a control for the ligand additions, especially for the growth factor signaling. So when we're adding EGF and HGF, which is going to activate ERK and AKT signaling, it's good to have that PBS condition to kind of know that those you're starting from a low or comparing to a low condition without those added growth factors. PBS had its own complications because that's going to end up with likely the fewest number of cells. And so in order to collect those samples, oftentimes at the 48-hour time point, we had to pool from multiple different wells, which I'll talk about in a second. So the next question we had to think about is the number of cells to plate, because this is a time course experiment, and also the plate format. So the picture in the lower left-hand corner here shows the EGF condition at 48 hours. And what we tried to do is, based on the number of cells here, we tried to work backwards because we didn't want the cells to be overly confluent. And so that at the 48-hour time point, specifically in the EGF condition, we'd begin to get other signals in the data from that confluence. So we tried to ensure that the EGF condition never got to a confluent state, and that dictated the number of cells that we started with. In terms of the plate format, we tried to design it so that it reduced the number of plates while maximizing the amount of material for each plate. So we ended up settling on an 8-well collagen-coated plates, which allowed us to test the 7 different conditions, and also allowed 2 different PBS conditions, which I talked about previously, ended up with relatively little cell material, requiring the need to pool across the different wells within there. In order to do the experiment and try to make sure that this 0-hour time point and the early time points weren't impacted significantly by the prior behavior of the cells, and specifically in growth factor signaling and also in cell proliferation, we did a media exchange. And so in the actual experiment, we plated cells in full growth media, which we found necessary in order for the cells to actually attach to the plate. Then 6 hours later, we did a PBS wash to remove kind of the growth factors. And then we did a media exchange where we used that experimental media, which was lacking in EGF and insulin within the media. The general idea then is that over the next 18 hours, cells that were in G1 would proliferate and then end up so that all the cells around time 0 would approximately be in this G1 state without any sort of growth factor signaling. In addition, we thought about the number of time points to collect and when we collected those time points. And in doing those, we're thinking about 2 different things. So one of the things was the timing of the molecular changes. Some changes in the biological signals you expect to occur relatively quickly, such as changes in signaling, and other changes you expect to be much slower, specifically changes in RNA-seq and also epigenetic changes. So for signaling, we're tended to collect the 0, 1, 4, 8, 24, and 48-hour time points, whereas for the attack-seq and also for the RNA-seq, we collected the 0, 24, and 48 hours. Additionally, we had to consider feasibility in collecting the samples. So for some of these sample type collections, it requires a large amount of hands-on time immediately after collecting the samples, which prevents collecting samples relatively close together. So in trying to get the plates prepared at 0 hours and then trying to collect a 1 hour, it didn't allow time period in, for example, for a 30-minute time point within there. So potentially we lose some of the kind of early signaling within there. Additionally, we considered extending the time period up to later time points. So for example, 72 hours. But again, we begin to be limited in the number of cells. So as I talked about previously, we set the number of cells based on the EGF condition and work back from there. And to go 72 hours so that we have another doubling of the cells, we'd have to significantly reduce the number of cells plated at the time zero time point. In addition, you start to run into issues that the media is changing over that time period. So either through, release of signals from the cells to condition the media, or depletion of the growth factors within the media. We tried to design the overall experiment so it was as robust as possible. In doing that, we collected a minimum of 3 different replicates for each of the samples that were collected. And then in analyzing the data, we found relatively high replicate to replicate correlation. I'm showing examples here of RPPA replicates. So comparing replicate A against B, A against C, and then B against C here. You can see those replicate correlations graphed on the panel on the right here, in addition to a kind of null where we're randomly matching antibody responses within there. So the data itself is relatively biological robust in the fact that we did collect multiple replicates, and those replicates are well aligned. In doing this experiment, we tried to collect as many different assay types as were possible. And in doing that, it required splitting the assay types across different collections. So we did 3 different collections. The cell culture for collections 1 and 2 were handled specifically by OHSU to try and minimize any kind of university to university variation, as Peter talked about earlier today. In addition, we weren't able to collect all these samples at a single collection because of the number of cells that are required for each different assay. So specifically, the GCP requires a large number of cells, whereas some of the other assays you can get by with relatively few. In addition, because we were plating so many cells, we had so many different samples and treatments, we tried to ensure that the experiment was never too complicated so that something would fail and we'd have to completely remove a replicate. So we've spent a lot of time trying to identify kind of the parameters needed to make the experiment go smoothly, and so we could actually collect all the samples that were necessary to make it as robust as possible. And on the next slide, just kind of showing an example. So for collection 1, this is for a single replicate. We would plate 19 different plates of cells. 15 of these were ligand treated plates. And you can see the relative division of how this was done. So we had a set of plates that were designed for RPPA, a set of plates for RNA-seq. We combined the ATAC-seq and L1000 and then split those samples in half for each of the assays. And we also had the IF plates. One of the things you notice here is that the early time points required doubling up of the plates and pooling across samples in order to get enough material for that assay. The GCP required significantly more cells, and so for the time 0 plate, we had to have 3 times 0 plates, and likewise, we had to have 3 plates for condition. And so this was a large number of cells that didn't allow us to collect the other assays at the same point. But as I'll talk about also on the next slide, we wanted to make sure that we had controls that were built in across the different collections. So for the GCP for collection 2, we also had L1000 as a kind of molecular readout, and then IF paired with it. And so this is the reason why we have so many L1000 samples across the different experiments. So we wanted to have both a molecular and then also a kind of behavioral phenotype so that we can make sure that the ligands were actually inducing response in each different replicate that we collected. So for collections 1, 2, and 3, we have IF and L1000 across. And so we can make comparisons to see if any of the kind of phenotypes or the molecular responses changed across any of the assays. So to summarize here, there's a lot of decision points that go into designing a robust experiment. So it took a lot of kind of preliminary experiments to optimize to make sure that once we actually dove in to plate all these cells and collect them, that things were likely to turn out, or were likely to find kind of responses within there. In addition, it took a lot of coordination across the different centers. So for collection 3, which was collected at HMS, there was a lot of coordination to make sure that how we were handling the cells was consistent with how they were handling the cells. And then overall, there's inherent trade-offs to each methodological decision. So we tried to think through what we lose by making what decision and what we potentially gain by making another. Overall, there were quite a few people who were involved, both at OHSU, but also at HMS, and then also at the different other link centers that helped in the analysis of the data. So with that, I'd be welcome to take any sort of questions that people have.
Great, thanks, Sean. I don't see any in the chat, and at the moment everyone is muted. So why don't we switch over to Mark to show about where these data are living, and then if people have questions about either any aspect of the presentation, we can take them at the end.
All right, did that pop up full screen?
Yes, perfect.
Okay, great, thanks. So I'm going to cover where the data is living, and this is the work of actually putting the data up there and organizing of Daniel, Derek, Ian McLean, Sean, Laura, and myself, along with help from Kenny Daly and Larson Almberg at Synapse. And so what I'm going to do is show mostly the live where the data is on Synapse, and on there what we're going to go through are the different levels of, we'll see raw, normalized, and median summarized data, and the metadata that explains, that captures all of what Sean was just describing, all the different experiment parameters and trade-offs, so that can go along with the data to be able to understand or deal with any batch effects and such. This is Synapse website coming up, the landing page here, and so links MCF10A molecular deep dive. MDD is some acronym that you'll see often in our code and file names to keep things straight. And what we've got is a simple explanation that as we finish off the paper that we're working on, this will get buffed out with a little bit more explanation. So what I'm going to do is kind of dive into where the data itself is. So by clicking on the button that says data files, we'll go to a version of it that is kind of a folder-based looking organization. So here we have the different assays that we've run, and Sean described the MIMA briefly. This was an assay that we ran before the others to be able to select the ligands, and then we have TAC-seq, cyclic IF, the GCP, global chromatin profiling, immunofluorescence, RPP, L1000, RPPA, and RNA-seq. So the metadata, let me go ahead and start there, and so the metadata has within it it's organized by specimen ID. So it's a unique identifier for every specimen that Sean and others had in the experiment. And so it has the ligands, the time point, which collection, what replicate in there. And so if we this is a file that from a bioinformatics standpoint is going to be key to be able to bring in that experimental data. So it's a file, this is a simple text file preview of it, where the specimen IDs have unique names, and then all of the information about that particular specimen can be joined to the data to be able to bring that along with the data itself. So we've done that with the data, and so I'm going to use the RPPA as an example. And here you can see that this is typical of how we've organized it, where we'll have an archive that may or may not have anything in it. The data itself, metadata, in a simple README file. I'm going to go into the data, and what we'll have in here is the level 0 data. In this case, the level 0 data is typically what we would get from the core, if there was a core involved, or if we did the experiment itself, say in the case of IF, the raw files that come in there. So these are files that would come from MD Anderson, who ran the RPPA. And for people that are used to processing RPPA data, these files are untouched, and they can roll directly into their pipelines. In addition, we've taken that data and we've created a level 3 version of that data. And here we've married the data from the core to our experimental metadata. And now the specimen IDs have all of the RPPA information married to them. And so these are, again, relatively easy to work with CSV files. But here, all of the replicate structure is in place. We talked about having 3 replicates. And so these are in this file as well. And while I'm here, let me show some information about this. So what the Synapse has the ability to show you the provenance of the file. So we're looking at this RPPA level 3 file down here. And what I can see is that it came from this file, and this one. So these are the 3 raw files that I was showing that came from the core. Here's our sample annotation file. And then here's a link to the GitHub repo that has the code. And in this case, it's a script called, it's written in R. It's a script called MBD RPPA preprocess. And it goes through and marries in essence, it's marrying the data and the metadata and doing a little bit of renaming in order to be able to get a clean CSV file to be able to work with. The next level up from that level 3 is a level 4, where now we've taken all of the replicates and summarized, median summarized across those replicates to be able to get data that is easier to use and more direct to use. So in here, every row for the RPPA is a protein that's been measured in that RPPA assay, and every column is a different condition that was run. So here it's the BMP2 condition at time 1, at hour 24, at hour 4, at 48. So these are, again, they're summarized CSV files that are relatively easy to use. And I'm gonna step back just a little bit here to talk about this data description area, because here we've got a description of the different data levels, and when we get down to the level 3 and level 4, the data has been normalized. It's at the replicate level. At these lower levels, because we're getting assays of different types, there's different processing and some slightly different definitions that need to fit in here. But we've laid out what we've done at these levels from 0, the rawest form, up to the median summarized for every ligand time point. So going back to the overview, so just to show a little bit, here are the other, the attack-seq, cyclic IF, and the rest of them are all very similar to the format that I just ran through for the RPPA. Level 0, here we got a level 1, but we also, the key ones that we think people would use to do their own processing would be the level 3 or level 4 data. But if you want to start at level 0, all the information is there. one other way to get at this data is through a synapse table. And the nice thing about this table is that it allows you to do some filtering. So here, if we say that I just want to see we'll go back for consistency, the RPPA files. I click on that, and now I've got all the RPPA files, whether they're data files or metadata files. And so if I say I want to see the level 3, what this little one here showing me that there's one level 1 file, there's one level 4 file, 3 level 3 files. The cell line is MCF10A. So this allows me to kind of filter down the data set to be able to find particular files that I may be interested in, and they can be additive. So if I were to say I want the cyclic IF data and the RPPA data, and I want the level 3 versions of those, there's 2 of them, and then I come over here and I can say add these to a download list. So I can go through and keep building up my download list, and then when I'm ready, I can go ahead and say download all of those, and Synapse will create a zip file and then do a download for it. So all of that kind of work is what I'm showing you is how to do it manually here, but there's also programmatic access to all of these files where, based on any of these metadata settings about the files, the annotations about the file, there can be a query against the Synapse API that will result in any selection. So what the level of the file, the assay type are all selectable and then downloaded directly by a script. And Synapse has support for Python, R, and command line scripting. So, and I think that's about it, and be happy to take any questions.
Thank you, Mark.
Sure.
I think the key thing is if people go to that web page, it's those big blue buttons on the top that they want to push in order to get to the data, right?
Right. Yes. These right here.
Excellent. Thank you.
Mm-hmm.
So we can open up the floor if there are any questions. I know it's a small and intimate group we have here today. So happy to take questions now, or of course to be available by email. If people go in to access the data and have questions, they can always email us with any questions about the data or the experimental design as well.
I had a quick question. Hi, everyone.
Hello.
Hi, so my name is Divya Murli, and I'm from Dr. Subramaniam's lab from UC San Diego. I had a quick question. I was wondering if the immunofluorescence data is also available on Synapse, like the images.
Right now, we posted the feature files.
Okay.
The images themselves will take a little bit of work to get together. Sharing image data is one of the big challenges across the field, but that's something we can work toward.
Okay. Thank you.
All right.
Okay.
Well, if there are no other questions, I would say we can close the session. Thanks, Sean and Mark, to both of you for putting together those very complete presentations. And thank you all for joining in.
Great. Thanks.
Thank you.
Thank you.